Good morning to one and all present. Welcome to a module on protein secondary prediction. I'm Dr. Sincy Joseph, Assistant Professor, Department of Botany, Nirmala College for Women, Coimbatore. So, the different level of protein structures shows that it has four structures, such as primary protein structure, secondary protein structure, tertiary, and quaternary structure, which is a little more complicated compared to the other three. The methods for protein structure prediction is generally classified into two types: experimental methods and computational methods. So, under experimental methods, we have three types, that is X-ray crystallography, NMR spectroscopy, and cryo method. Under computational methods, we have homology modeling, fold recognition, and ab initio modeling. The experimental protein structure determination is determined by three methods, such as X-ray crystallography, NMR, and electron microscopy. The computational methods involves major techniques such as template modeling, homology modeling, threading, and both is used for to identify a known protein structure. And the template-free modeling and ab initio methods, which is based on knowledge, is used without knowing the protein structure. The homology modeling is also known as comparative modeling, where the protein prediction structure is based on sequence homology with known structure. So, the principle behind this is if two proteins share a high enough sequence similarity, they are likely to have similar three-dimensional structures. So, the basic modeling servers are mod-based, Swiss model, etc., and it also fails in logos. So the six steps that is involved is first is the template selection, second is the sequence alignment, third is the model building, which involves backbone and model building and loop modeling, and the fourth one is side chain refinement, and fifth one is model refinement, sixth one is model evaluation. So it has been observed that even proteins with 30% sequence identity fold into similar structures. And it also doesn't work for remote homologs, that is, having lesser identity, that is, the percentage identity is below 30. So this image shows how it works, how to find a template, align a target sequence, and generate the model and refine the model. Threading is given sequence of a protein P with unknown structure and the database of fold of known folds. So we have to find the possible fold for P and evaluate the quality of such arrangements. So we place the residues of unknown P along the backbone of a known structure and determine the stability of side chains in that arrangement. We can predict the structural fold of unknown protein sequences by fitting the sequence into a structural database and selecting the best fitting fold. We can identify the structurally similar proteins even without detectable sequences similarity using the two algorithms such as pairwise energy based method and profile. In pairwise energy based method, which is also known as threading, we search for a structural fold database to find the best matching structural fold using energy based criteria. And we use dynamic programming and heuristic approaches. We calculate the energy for raw model, and the lowest energy fold corresponds to to a structurally group of the compactable fold. For prime profile based method, a profile is constructed for related protein structures. It is generated by superimposition of the structure to expose corresponding residues. The second structure type, polarity, hydrophobicity, and the protein fold which is to be predicted does not exist in the fold library. Then the method will be failed. This diagram represents the various profile matchings, the threading process, which includes the prediction based threading profile and profile matching. Ab initio protein structure prediction. It is built 30 protein 3D structures from sequence based on physical principles. The importance is they can even used in computation. It is high, it has a high computational demanding, and it is based on the physical models. They are indispensable complementary methods to knowledge based approach, and knowledge based approach will fail if the structure homologous is not available or possible undiscovered new fold. Rosetta is a web server for protein 3D structure, and this shows the diagrammatic representation of various protein structure prediction. And this diagram shows the overall approach of a protein sequence and how do we conclude with a 3D protein model. Thank you. Thank you for watching the video.